For today's discussion, you're no doubt aware that before the pandemic, an estimated 1/3 of a billion people suffered from depression, 2/3 are undiagnosed, and 1/3 of those who are diagnosed are treatment resistant. And the Global Burden of Diseases study revealed that mental health disorders are among the top 10 leading causes of disability worldwide, with no evidence of a global reduction in that disease burden since 1990, actually. And there's been a substantial increase in the prevalence and burden of major depressive disorder and anxiety disorders as a result of the COVID-19 pandemic. In addition to that, we're all acutely aware, of course, of the opioid crisis in this country that tragically takes over 100 people per day in this country, and the steady and alarming rise in suicide rate over the past 2 decades. So this wholly inadequate state of affairs and relative lack of progress in therapeutics makes today's session really that much more important. To say that we're in the midst of a psychedelic therapeutic revolution would perhaps now be an understatement. The nation's top universities, including UCSF and Hopkins and Yale and Berkeley and Mount Sinai and others, are establishing psychedelic research centers. Investors are pouring money into more than a dozen startups. And states and cities across the country are beginning to loosen restrictions on these drugs. I think the first steps, actually, in what some hope will lead to federal decriminalization of psychedelics for therapeutic use. So today we're very lucky to have one of the world's foremost experts and a colleague of Professor Nutt's to expand on this important discussion and provide us with an overview of the biological underpinnings of these disorders and the mechanism of action of psychedelics. Dr. Carhart-Harris is the Ralph Metzner Distinguished Professor of Neurology and Psychiatry at the University of California in San Francisco. He is the founding director of the UCSF Neuroscape Psychedelics Division. Previously, he led the Center for Psychedelic Research at Imperial College of London. He has designed a number of functional brain imaging studies with psilocybin, LSD, MDMA, and DMT, and completed multiple clinical trials of psilocybin for depression. Dr. Carhart-Harris is one of the most cited researchers in the world of psychedelic science, helping to elucidate the neural and biochemical underpinnings of psychedelic molecules and their therapeutic effects. I'm just so delighted that he's taken the time to be with us today. Dr. Carhart-Harris, thank you, welcome, and we really look forward to your presentation today.
Thank you, David. I'm delighted to be here. I really hope I can give everyone an engaging presentation on this topic, a particular passion of mine, trying to understand how psychedelic compounds work in the brain and how they could be utilized for therapeutic purposes. My research really focuses on the classic psychedelics, and one way we can work towards a clearer, more precise definition of these compounds is to look at their pharmacology. Just to rewind a little bit, the etymology of psychedelic, soul-revealing compounds, is just that this term is meant to denote what then was considered the defining property of them, as abstract as it sounds, that these are compounds that reveal the psyche, the soul, if you want. But be that as it may, it's in a sense CRISPR science to look at the pharmacology, the proteomic and molecular beginnings of that sort of downstream action. And when we do that, we discover that actually there is a certain neurotransmitter receptor that seems to be key to the action, the signature action of these classic psychedelic compounds. That receptor is the serotonin 2A receptor, one of at least 14 different serotonin receptor types. We can see where it's expressed in the human brain, in the living human brain. So this map, PET imaging map that we're looking at, is arguably the most informative mapping of where these receptors are in the living human brain, we can see that the receptor is very much a cortical receptor, high expression, mostly throughout the cortex, but it's not entirely homogeneous. What you might be able to see is that there's especially high expression in high-level cortex, actually cortex that is most recent, you might say, evolutionarily, but also developmentally. This is cortex that expands disproportionately as we develop into adulthood, and has expanded disproportionately as we've evolved into human beings, if you want. So hierarchically, high-level cortex is where this key receptor for the action of classic psychedelics is housed. How do we know that this receptor is critical to the action of classic psychedelics? Well, I suppose the story began in the mid-1980s when it was discovered that there was quite a tight relationship between the affinity of classic psychedelics for the serotonin 2A receptor and the potency of a range of different psychedelics. So the higher the affinity, the higher the binding potential or stickiness, if you want, for the serotonin 2A receptor specifically, the higher the potency. So, for example, LSD, very high affinity for the serotonin 2A receptor and incredibly potent, psychoactive in microgram doses. We think something in the ballpark of, say, 15 micrograms. It is about the dosage at which people would feel the psychedelic effects of LSD, but you compare that to mescaline, where a typical dosage is in the range of, say, 400 milligrams is, for example, what Aldous Huxley took when he wrote his famous Doors of Perception on his experience with pure mescaline. He took 400 milligrams there. So, you know, quite a difference, and that relates to the affinity for the 2A receptor, relatively lower with the compound mescaline, much higher affinity with LSD, and that translates into the potency. Now, since then, another staple finding, really a pivotal finding in human psychedelic research, came from Franz Vollenweider, where he found that pre-treating with a somewhat selective, not entirely, but we'll get there, serotonin 2A receptor blocker or antagonist, if you give that before a psychedelic, in this case psilocybin is where he began his work, the active compound, or at least the prodrug of the active compound in so-called magic mushrooms, psilocybe mushrooms. So he would give the blocker before giving the psychedelic, blocking the 2A receptor somewhat selectively, and the signature psychological effects of the psychedelic would not be felt. They would be significantly attenuated, if not entirely abolished. Now, that finding of pre-treatment with a 2A blocker or antagonist blocking the effects of a psychedelic is very well replicated now in humans, which is critical, and then in non-human animals, looking at behavioral indices of psychedelic action. And just to kind of put an extra stamp of authority on the role of the 2A receptor, it's also been found now using PET imaging that if you displace a ligand binding very selectively actually for the serotonin 2A receptor with a range of dosages of psilocybin, you give the psilocybin, it displaces what we call the hot ligand, the radioactively labeled, highly selective 2A agonist receptor ligand, binding to serotonin 2A receptors in their functionally available state, they're available for signaling if you want. So you give the psilocybin, it's now competing for the hot ligand, displacing it away, and we can look at the proportion of 2A receptors that are occupied by different dosages of psilocybin. So this is the chart being shown here. This is work out of Gita Knudsen's lab in Copenhagen. And we see what we expected, really, that there is a relationship between the occupancy of 2A receptors by different dosages of psilocybin and the subjective intensity of effects, psychological effects produced by those different dosages. So the higher the occupancy, the more 2A receptors bound to by the psilocybin, the more intense the psychedelic effects. Now, it was a small sample, as is typical with PET imaging. There are hints at some non-linearity in the relationship. So, you get above a certain inflection point in 2A binding, looking at the chart here, around about sort of 55-60% binding to 2A, and there seems to be an inflection in the subjective intensity of effects produced by such a dosage of psilocybin. Time will tell if that non-linearity is a principle. If it is, it would make somewhat intuitive sense. And people should know that the dosages that are achieving this level of occupancy, 60% plus, are in the dose range of psilocybin that are typically used today in the clinical trials of psilocybin therapy for, say, depression, anorexia, a number of different indications being looked at now. So there could be a critical dosage range to achieve the desirable level of intensity in subjective effects. And we'll get to why that's important as we move forward, but perhaps the early spoiler is that there seems to be quite a robust and reliable relationship between the quality of the psychedelic experience and the therapeutic effects that are seen subsequently. And we think that that's likely to be very important. So you've heard that the serotonin 2A receptor is where it all begins, but now we should ask ourselves the question, what does it do? What does serotonin 2A receptor signaling achieve? What's its function? And that can link up with its evolutionary function. Why is it there? Why do we have the function that's activated by 2A receptor activation? And when we look at the properties, the effects of 2A signaling, we can see that there's a number of different properties that could fall underneath an umbrella construct of plasticity, the ability of a phenomenon, a system, a property, a phenotype to be shaped or molded. Look it up in the dictionary. That's what plasticity is. It's not change per se, although it's related, and neither is it growth per se, but it's related. More precisely, plasticity is the ability to be shaped or molded. So you can see how it links in to, for example, learning. So let's run through some of these properties and the evidences that are cited. Yes, we can look at neuroplasticity, changes in the actual anatomy, very tangible changes, looking at neuronal anatomy, synaptic anatomy. And if we see changes there with the application of psychedelics, which is pretty well demonstrated now, you can see the schematic on the right with a range of different 2A agonist classic psychedelics, increasing synaptogenesis, so anatomically increasing the density of components of the synapse, synaptic boutons, for example, developing with the application of these drugs. So this is, very tangible change in neuronal anatomy, which is perhaps one of the sort of favorite markers of neuroplasticity that is being seen with psychedelics. This effect seems to be somewhat cortically specific. We can see this from an older study, albeit looking at a different marker of neuroplasticity, brain-derived neurotrophic factor, BDNF, and you can see in the parietal cortex there of rodents this near doubling of BDNF expression. And it's also worth seeing that that rule of an increase in neuroplasticity isn't true throughout the brain. Look at the dentate gyrus there in the hippocampus, an actual decrease in BDNF there. So this increase in neuroplasticity does actually seem to be a somewhat specific property to the cortex, cortical neuroplasticity. And remember where those 2A receptors are. They're highly expressed in cortex. So that holds up. Now, that synaptogenesis work was done in vitro, and now we have this important finding of similar changes, but in vivo. What's exciting about this finding is, yes, that it's in vivo, but also that with a single application of psilocybin, the increases in synaptogenesis, spine density we're looking at here, was protracted. It was enduring. It was actually seen to persist out to over a month after that initial single dose. Now, that's exciting and makes sense because we know from the human clinical trials that even a single administration of psilocybin can have enduring psychological effects, changing, perhaps transforming certain psychological phenotypes, personality traits, and therapeutic outcomes as well. So this finding in non-human animals of an increase in neuroplasticity that's enduring matches that. So I think it's part of the story. It's really the biological part of the story. And as we go on, you'll hear my perspective that psychedelic therapy is a combination treatment. It's the combination of a biological action with a contextual action as well. So we could look at some other properties. I'll just run through these a little more swiftly. We can look at brain development. I've highlighted a particular study there that looked at embryonic brain in a variety of different species and found that 2A signaling in this very early brain, pre-birth, was increasing the proliferation of what you might call pre-neurons, these basal progenitor cells that are very important cells for the development of neurons, cortical neurons, and seem to be related to cortical expansion, which of course is so marked in the human species. And because of that, and because the relationship seemed to be somewhat specific to 2A signaling, the authors in the abstract of this paper published in Neuron make the extrapolation that perhaps 2A signaling has been involved in the evolution of the human brain, that this is a mechanism that is necessary for cortical expansion. Of course, what sets apart our brains from other animals is the sheer size of cortex, especially relative to body mass. So there we have that, and then we can look at learning, and actually I think this is somewhat of a low-hanging fruit for research going forwards. It's more of a concentrated look at changes in learning with the application of psychedelics. In rabbits, some important work has been done looking at the acceleration of learning rate. And actually learning rate, I think, is a particularly juicy, low-hanging fruit to look at. Here with LSD, you can see those open circles. I think they're circles. That's the curve where learning, this is low-level associative learning in rabbits, aversive learning. It was a response to air puffs to the eye. That being accelerated with LSD. And if we look at the lower curve of the 4, that's with the application of a 2A blocker, slowing down, or if you want, retarding learning, this low-level learning. So a role for the 2A receptor in learning rate. And why could this be important? Well, in a sense, we're getting closer to the true definition of plasticity here, the ability to be shaped or molded. And what we're looking at is a catalyzing of a behavioral change, speeding up of the ability to be shaped or molded with LSD here. So this could be, close to the money, if you want, of what psychedelics are doing behaviorally, and is probably the property that is harnessed in psychedelic therapy, this combination treatment. And we can look in humans at varieties of context sensitivity, if you want. We could look at cognitive flexibility post-application of a psychedelic. The evidence is somewhat variable if we look at the acute action, but look post-acute, you'll see increases in cognitive flexibility, reversal learning, the ability to shift your behavior after learning a rule. Rule changes and you can shift more easily with an increase in cognitive flexibility. We could also look up a level of abstraction to psychological flexibility, a construct that has been somewhat popularized in relation to what you might call third wave psychotherapies, acceptance and commitment therapy, perhaps most specifically mindfulness-based cognitive therapy as well. So that ability to sit with all emotions and feeling states, to accept rather than seek some kind of avoidant defense mechanism that might then reinforce and become rigid in nature. So that ability to remain open and flexible, that's what is being referred to and measured by psychological flexibility, increases in that. And then we could look at environmental sensitivity. The two examples I cite here are sensitivity to music, the emotional impact of music, and sensitivity to a hypnotic suggestibility paradigm. A couple of studies from myself and my colleagues, increases in those effects with psychedelics, applications of psychedelics to humans. So now let's reflect for a moment on how this might tie in with the evolution of the 2A receptor and its functioning. And what I discovered as I was looking at conditions in which 2A receptor functioning and availability is changed, I was finding that there seemed to be an association with stress. And diving deeper into the data, and more critically, it became evident that chronic stress in particular was associated with an upregulation of both 2A availability and also its sensitivity to signaling, whatever that readout is of 2A signaling. And so, for example, hypoxia has been associated with a marked increase in 2A availability, and also we can look at readout of 2A signaling with chronic stress. Here, wet dog shakes, head shaking is a behavioral readout of the action of 2A agonist psychedelics, increased in the context of chronic stress. And then we also have another study cited here, maternal separation and chronic immobilization stress, and this massive increase in readout of 2A signaling, this early plasticity gene arc. So those are just three examples, but there's a lot of studies cited there. And it seems to be chronic stress and, in a sense, deprivation, conditions of deprivation, social deprivation in animals, seems to upregulate, if you want, prime or sensitize the 2A system. And then acute stress is arguably the most reliable way to release serotonin short of giving a drug. So 3 different varieties of, say, punishment paradigms here. We have tail pinch, handling, and swim stress, all markedly increasing serotonin release. So here you have the double whammy, of course, of a primed 2A system, and then the release of the endogenous ligand, serotonin, onto that primed system is going to engage, if you want, naturally, albeit under conditions of adversity, stress, activating the 2A system, as you've just heard in the previous slide, a system that engages, that activates plasticity, the ability to be shaped or molded. So here we can, in a sense, see the formula and answer the question of why we have the 2A receptor. And the inference that follows is that it is involved in adaptability under conditions of adversity. So that, to me, makes sense that this is the system that classic psychedelics hijack. And what they do is to, in a very marked way, really engage this plasticity, adaptability system, and they probably, are hijacking a system for rapid adaptability, perhaps deep and rapid adaptability or change. For example, to circumstances of adversity, of hardship, where a change is needed, perhaps life or death, it's needed. This change is essential for survival. Here's just a bit of evidence to link stress itself to cortical increases in a plasticity marker, acute stress at least. So all of that background helped motivate this construct that I came up with the help of a colleague, Ari Brouwer, of pivotal mental states. And for me, this is a construct that can help us understand what psychedelic therapy is, what it's doing, and also what's essential for it to work well. And the construct can be defined as states of transient hyperplasticity in which the organism is especially sensitive to their contextual conditions. They're highly sensitive people. They're highly susceptible to environmental conditions. If those conditions are adverse and they're in this highly plastic state, then they could be pushed in a particular direction that could potentially be pathological. You could imagine that being a formula that could, for example, precipitate a psychotic disorder, or PTSD, for example, post-traumatic stress disorder, hardship twinned with plasticity, enhanced learning, and the learning is, in a sense, maladaptive. In another sense, you could say, adaptive. But that's another story. But think of another condition, that transient hyperplasticity engaged by 2A receptor agonism, twinned with a compassionate, solid and trustworthy therapeutic container. And that's going to be potentially a formula for positive therapeutic change. And I would say that is the formula that we're trying to harness and engage with this combination treatment that is psychedelic therapy. So then if you look at the studies, the modern human trials that have looked at the effects of psychedelics on different mental health outcomes, then you'll notice a few things. You'll notice that all of these studies have manipulated the context in a positive way. I think all of these studies have involved music listening. It's a detail that could easily be overlooked, but let's not do that, Often the conditions are lie back on, say, a comfortable sofa with two mental health professionals who you've built a sense of trust with. The typical formula is 2. These days it could be 1 with an overseeing sort of supervisor. And eye shade might go on, and music listening through headphones, and perhaps also in adjacent speakers playing into the room. So this is a very supportive environment, and one shouldn't gloss over the psychological preparation that's gone into the mix ahead of the experience, and then the psychological support, debriefing and integration work, we call it, that happens after the experience as well. So this is very extensive psychotherapy, something that could be critiqued, for how realistic is it that you could have such a level of extensive therapy going forwards. But that is the formula, that is what's been done, and that seems to be the formula that has produced these exciting findings. So it's twinning this hyperplastic state induced by 2A agonism with these very supportive contextual conditions is reaping these very promising findings. You can see that black bar, that average of the patients in our treatment-resistant depression trial, and those rapid drops at 1 week that are relatively sustained. But also note the variance, and that's another thing to consider, those cases where you might not see the rule of the drop in symptom severity. So another principle is that rapid action, the enduring action, and the last one to draw your attention to, highlighted in the red font, is this potential transdiagnostic action. I would say these properties, the rapid, enduring, and transdiagnostic action, are probably what's, so promising about psychedelic therapy potentially. And just different indications and aspects of mental health being cited here: well-being in healthy volunteers, end-of-life distress, demoralization in AIDS survivors, that study, different addictions, depression studies, growing evidence there, and obsessive-compulsive disorder. This is a Vanguard series, so you should also know we're getting very good readouts in anorexia with psilocybin therapy. This is work with my colleagues at Imperial College London, other teams looking at a variety of different indications. So while we have what's published, do expect in the next few years really quite a broad variety of different indications, and I believe some impressive findings are going to be seen in these studies that are being carried out as we speak. So in order to understand a solution to a problem, it's necessary to understand the problem itself first in some depth. And based on that principle, we could ask ourselves the question, is there a common denominator to a range of different psychopathologies? And perhaps that common denominator is being targeted by this potential transdiagnostically active intervention. And there I just want to put forward the hypothesis that there is a common space, and it is the development of patterns of thinking and behavior that in a sense are maladaptive, in another sense you could say are adaptive. It's not really disorder, but rather an alternative order in these psychopathologies, whether it's the negative cognitive bias in depression or symptoms of post-traumatic stress disorder, symptoms of addiction, specific ways of looking at the self in eating and image disorders, specific behaviors that become reinforced and entrenched in OCD. And so what I'm suggesting here is that this is a learning process. In a sense, the state space for mind and behavior has become constrained to a particular space. In depression, say, it's a negatively biased space. In addictions, it's the behavior and thinking around whatever the preferred objects of addiction are. Let's say it's a drug, methamphetamine, for example. You sort of become an expert in that drug, and your whole being is now concentrated, focused around that selective state space. So it's a process of reinforcement. In evolutionary science, there's this construct of canalization. In a sense, it's the opposite of plasticity. It's a reinforcement of a particular phenotype that gets stamped in, entrenched, rather than a phenotype that is open for being shaped or molded. And so that canalization process is what I'm suggesting is a common space that is underlying a range of different psychopathologies and is the target of psychedelic therapy. One might say, any really deeply effective treatment should target this common space. I put bad habits in inverted commas. I should probably put bad in inverted commas as well, because, for some people, an addiction out of a background of intense trauma and adversity is doing something for them, and to try and understand that and appreciate that, I think will help us better understand how to find a solution to that problem, and to provide that solution in the most compassionate way if you understand how the alternative order, not the disorder, the alternative order has been serving the patient. So that's how I come at things. Psychologically supported plasticity with psychedelic therapy, a combination treatment. So that was a lot of background, but I think it's necessary because it is working towards that thorough understanding, or at least a hypothesis, a model of the therapeutic action of psychedelics, classic psychedelic therapy. What happens when you leave the trenches with the acute action of psychedelics? Well, let's go there now, looking at acute functional brain imaging. And the principle that emerges is one of a decrease in segregation in the brain, a decrease in modularity. We can see that here with psilocybin, where different modules are talking to each other more freely under the action of psilocybin there relative to placebo. We can look at this in a slightly different way, seeding the primary visual cortex, actually done in this study with retinotopic mapping, so this is V1, and then asking where in the brain is activity coupled to V1, where's the activity synchronous, and we see that ordinarily it's other regions of the visual system, but under LSD it's most of the brain is now coupled in its fluctuating activity with V1. So there's a breakdown in this modularity, there's a breakdown of boundaries, and this global integration that you see under psychedelics. This was scanning with eyes closed, and in this particular data set the effect correlated under LSD with the intensity of complex visual imagery with eyes closed. So it seems to relate to the phenomenology, it seems to relate to the subjective effects. Here with DMT and other compounds, same effect, increase in global integration in these regions, labeled with that hot color there. So it seems to be a rule, and we can dive down into the rule and look at its dynamics. Here we're looking at different states over time. These are just the array of possible states that the brain can gravitate into at any particular moment. And what you see is that state on the far left is a globally coherent or integrated state that is visited disproportionately so under the psychedelic, in this case DMT, but we've found this with psilocybin and LSD also. And what are the states that are diminished in how often they're visited? Well, it turns out these are high-level systems, high-level networks. They include the default mode network, but not just the default mode network, other frontoparietal systems. And these are high-level systems. Hierarchically, they are high-level. They're involved in transmodal functions, so a lot of different things, hence transmodal across modalities. And they're also regions that house the 2A receptor to a disproportionate extent, and are regions that have expanded most in our species. And these are the systems, the states that are diminished in their visitation time under drug as their boundaries break down and the brain becomes more globally coherent or integrous in its activity. So another way to look at this, we can look at the principal gradient, which is essentially how the functional connectivity in the brain is organized ordinarily, and what's the main dimension of that organization. Or when that's been done, this quite famous work from Daniel Margulies, it separates unimodal cortex, visual cortex and motor cortex, from the transmodal high-level cortex. That's the principal gradient. You can see it clearly in that placebo PCB top left image, that sharp demarcation where the gradient is a dimension or an axis, is very clear. the one end you have the blue unimodal, and at the other end you have the yellow, the hot transmodal. So what you get under psychedelics, you get a collapse or a condensing of that gradient as these modules talk to each other more freely, more easily. This, I believe, will emerge as a very important principle of the action of psychedelics on the brain. Time will tell whether it's a signature action. I tend to think it might be, and there's a new paper that's just come out in Science Advances which speaks to this effect as well. Let's hope to get this work into the public domain. It's in preprint at the moment, has been stuck in review for a while, but let's hope it breaks out of that and gets to people's eyes very soon. It's there in preprint. Girn et al., Girn, is where to look. So that's the acute action of psychedelics on brain function. What about the post-acute action? So here we're looking at data in a population of people with depression, at least at baseline. Two studies, one on the left, one on the right. The first study is in treatment-resistant depression. The one on the right is in major depressive disorder, so depression more broadly defined. The intervention is psilocybin therapy, principally in red. And the effect, first of all, the metric that we're looking at is brain modularity, so how segregated is the brain, and the effect seen with psychedelic therapy post-treatment. In the first case, TRD trial, 1 day post-treatment. In the second case, MDD, 3 weeks after the second of 2 high-dose psilocybin therapy sessions. And the effect is a decrease in modularity, a decrease in the segregation of brain networks in the brain post-treatment. Now, if you're sharp, you might think, well, this looks a bit like what you were just talking about in terms of a increase in global brain integrity or a decrease in modularity on drug. And that is how we're interpreting this, that this is a kind of carryover effect of the acute action. And if the acute action could be characterized as a sort of freeing up of brain dynamics, then we're seeing a carryover of that post-acutely, at least in depression. Now, what's intriguing is that this effect correlated with improvements in symptom severity in both studies. In fact, it was predictive of improvements as well, longer-term improvements. So it seems to be a sort of desirable action of psilocybin therapy in depression. Bigger this effect, the bigger the improvement in the depression. So that's exciting, reliable at least across 2 studies, robust in 2 studies, and also a suggestion of some degree of specificity in that we did not see this effect with a conventional antidepressant drug, in this case escitalopram, a highly selective serotonin reuptake inhibitor that was taken every day up to pretty solid dose, 20 milligrams every day, over a 6-week period. So that did not change brain modularity, and neither was there a relationship with any changes in symptom severity with that particular intervention. So here we have this interesting principle, and apologies for the simplicity of this model until I can find someone to make me a nice energy landscape that's not pieces of paper. But the principle I just want to articulate is one of going from a state space that is relatively free, that I would argue characterizes the healthy brain, the ability to move around, to explore the kind of properties that you might see in a young brain, an infant brain. And then over time, through maturation, the landscape becomes more specialized as modularity increases, as we become specialists and our brains mature, we can speak and move around in a finessed way. But that could overshoot in psychopathology, like in depression, where there's a particular state space that characterizes the depression and negative rumination that becomes too deep as an energy well, and you get stuck in this rut. It's easy to fall into it. and hard to get out of it. And that's the kind of pathological state space, alternative order that I think psychedelic therapy could target. And through this carryover effect on decreasing brain modularity, I understand that as a kind of reflattening of the state space, so that those freer dynamics are promoted with successful psychedelic therapy. You can explore again, you can come out of your dark room and start to experience the world again. If we understand that model, you can also appreciate how brave it is for patients to come out of that dark room if you want, and also how difficult it could be. And so that process needs to be annexed with good psychological support. So we're moving towards, I would say, the final third or quarter of this talk now, and this is really summary material. Here we're looking at a range of different properties of the human brain, a reminder of where the 2A receptor is in this transmodal cortex principally, and then we can see, we'll move left to right, top and then bottom. We can see a map of macaque brain to human brain cortical expansion. Look how similar that pattern is with the 2A receptor. Hot colors in expanded cortex, hot colors showing us where the 2A receptor is. Look further along, top right, postnatal cortex expansion. This is expansion as we go from infants to adulthood. Similar pattern again. Let's look at the principal gradient. Similar pattern again, separating transmodal hot from blue unimodal. Let's look at connection length, longer fibers in this pattern. I'll call the pattern the human system, given that it corresponds with cortex that's expanded most in our species. We can look at between subject variability in the brain and see that there's more variability in this human system. It's a more open plastic system, open for shaping. We can look at myelination, the cabling, the insulation of fibers in the brain, lighter myelination in this human system, this plastic human system. We can look at information processing, more abstract information processed by, I'm moving bottom now, left to right, more abstract information processed by this human system. Longer temporal windows, richer information in those longer temporal windows processed by the human system. A certain style of metabolism that's fast, somewhat inefficient, but rapid, fast, corresponding to this pattern. And then just a reminder, if we look at changes in functional connectivity in the brain with a psychedelic, corresponding again to this transmodal human system. So what's the principle there? I would say it's this. It's that a fundamental property of human brain organization is being hit by classic psychedelics. That may well be why the classic psychedelic experience is so profoundly unusual as an altered state of consciousness, and perhaps why the state is so powerful as a potential, therapeutic mediator of change. Now, this model, Rebus, is an acronym for Relaxed Beliefs Under Psychedelics. It's done phonetically. And the principle is this: it borrows from a dominant model in cognitive neuroscience, hierarchical predictive processing, to say that which very briefly is a principle that our minds and brains have developed to be models, predictive models of the environments that they exist in. Now, models are held with differential degrees of confidence, precision weighting. And if they're organized hierarchically, you could alter precision weighting at different levels. And I'm saying with psychedelics, they principally target the precision weighting, the confidence of high-level models or beliefs, and they dial it down through their action, which is probably related to a dysregulation at a population, neural population level, entropic action, I call it. They'll dial down the precision weighting of these high-level internal models, relaxed beliefs under psychedelics. some optical illusions speaking to perceptual assumptions. We see something like this, and for some of you, you might be hallucinating some motion that doesn't actually exist because it's, in a sense, tickling your predictive models that say there should be motion here. There's not, but your models are dominating your perceptual experience if you're having that hallucination of motion. So how do you test something like that? Well, there's lots of different ways you could do it, because if hierarchical predictive processing is true as a principle, it also probably applies throughout the brain in multiple different ways. Different properties of brain organization should adhere to this principle of hierarchical predictive processing. So here we've selected just one for which there is some evidence that it's involved in predictive mechanisms, and it's traveling waves. You drop a pebble in a still pool, you'll see waves move in their directionality from where the pebble was dropped out to the periphery. Those are traveling waves, and you can pick them up in the cortex here with EEG, and you can see their directionality and how it changes with different circumstances. If you go from eyes open, where the information travels bottom up from the visual cortex up the hierarchy to the frontal cortex, you close your eyes, the directionality flips to a top-down dominance from the front to the back. So what happens this was our question. If you have your eyes closed and the direction of the traveling waves is top down, front to back, nothing to see here, and you inject someone with DMT, highly, profound psychedelic in its effects, we give a bolus of that, people have their eyes closed, what will we see in the traveling waves? We predicted we would see a flipping of those traveling waves as if the lights come on and you're seeing things. People did keep their eyes closed. And we saw it very clearly. DMT injection, the top-down traveling waves dip down, and bottom-up traveling waves come up. Very similar effect to what you see with visual stimulation, off and then on. Same pattern. So here, the principle, I would say, is supported with, you can articulate it in a simple way of less top-down being synonymous with a decrease in precision weighting of the high-level models and increase in bottom-up. And on a more general level, I think it could potentially explain a lot of the phenomenology of the psychedelic experience, phenomena like ego dissolution, the disintegration of the sense of self, if the self is a particularly high-level or deep internal model of the world and the agent in the world. So that drops away under psychedelics, often described by people and measured and shown across studies, very robust, dose-dependent ego dissolution. But also emergent phenomena, the rich visual experience, the theatre, complex animated theatre of activity that can play out in the mind's eye under a psychedelic, often very vivid and archetypal and emotionally loaded material, sometimes personal, sometimes transpersonal, that plays out under psychedelics, and the emotion, the strong emotion and personal memories that can come up as well. So this is sort of bottom-up information allowed to precipitate, allowed to emerge under psychedelics. I think this Rebus model helps explain that action. So I'll end now on a summary of a recent trial that we performed at Imperial College London, which was a head-to-head double-blind randomized control trial comparing psilocybin therapy, 2 sessions, 25 milligrams, 3 weeks apart, with 6 weeks of escitalopram, daily escitalopram. We matched for everything in each condition, in terms of taking placebo capsules every day in the psilocybin therapy condition to match for taking daily capsules with escitalopram. And in the escitalopram condition, we also had the same level of psychological support and 2 dosing sessions where everyone in the trial was told they would get psilocybin, but in the escitalopram group, those dosing sessions with psilocybin only involved 1 milligram of psilocybin, which we presume to have negligible activity. In the psilocybin condition, the red condition, it was 25 milligrams, so 25 times the dose, and that's a pretty big dose of psilocybin there. So that was the design, that head-to-head, fully randomized and blinded. And here are the outcomes. This is a forest plot where the two bars have to cross the midline in order to be greater than 95% confident of a significant difference between the two conditions. These are all our main efficacy outcomes. We've got 4 depression rating scales at the top, and then other varieties of mental health outcome. And a lot of green bars there, which significantly favor one of the conditions. It's the psilocybin condition. So it looks very promising for psilocybin. The one example that missed, that didn't significantly favor psilocybin, was actually the primary, pre-registered primary, at least depression primary outcome measure, the QIDS. So I would say, judge the data fairly, make the inferences that make sense to you. It missed on the primary, but hit on a broad range of other efficacy outcome measures. So I think a fair thing to say is it looks promising for psilocybin therapy versus a gold standard treatment for depression. Faster onset with psilocybin than the escitalopram. And then when we dived into the issue of those who came off medication to come into the trial because they were medicated with antidepressants, versus those who weren't medicated, they had depression, but they weren't on any psychiatric medication. We split the sample in that way, and you can see that split. On the left is the version of those individuals who weren't on any medication coming into the trial. So they didn't come off any medication because they weren't on anything. There we saw, even in the QIDS, significant separation at every time point. Very clear separation in favor of psilocybin, the red, bigger drops in depression scores there. So had we had, in a sense, a clean sample of people not on any psychiatric medication, it would have been very strong evidence in favor of the superiority of psilocybin therapy for depression versus a conventional antidepressant drug. But in the sample, we'd had about a third of the sample who were on some conventional antidepressant medication coming in, who had to come off. The washout period was 2 weeks, and from what we've seen, the suggestion is it wasn't long enough because it seemed to attenuate the difference between the two conditions. It affected the escitalopram condition more, where those who came off and went, in a sense, back on a conventional antidepressant did particularly well. And those in the psilocybin condition, was a slightly attenuated, I think it actually reached significance. So it was an attenuated response to the psilocybin as well. So this could speak to a number of different things: a sample bias, those who know meds and then go on. By that I mean conventional meds, go on a conventional med, do okay, they're less biased perhaps against it. Maybe there's something about discontinuation, dare I say discontinuation syndrome. You come off a conventional med, you don't feel so great, you go back on it, back to where I want to be, So there could be a few factors going on. There could be 2A downregulation in those who come off conventional meds, where the psilocybin has less of an effect because the 2A receptor is less available because of being on meds, downregulating 2A. So a few different reasons, but I think it's important to see these data because they could suggest that, for example, that miss on the primary, which actually dominated the reporting of this trial, might have been confounded by antidepressant discontinuation in a subsample. So I'll park it there. I hope you enjoyed that. Very happy now to take any questions, and thank you again for your attention.
You teased us in the beginning by saying that as many applications that are emerging as potential candidates for psychedelic therapy, there are many more to come. And so hopefully we can hold you to coming back to speak with us in the future and give us another update on how far we progress perhaps over the next 12 months or so. So we hope we can bring you back. This was absolutely outstanding. We really appreciate your time. Just thanks so much from all of us.